Cell migration is the process by which cells move from one location to another. Cell migration is essential for development and maintenance of multicellular organisms, like us. It is also vital in immune responses and pathological conditions. Scientists and engineers around the world are constantly looking to study cell migration in a 3D environment. Such research will lead to better understanding of cancer metastasis, drug diffusion through tissues, and the effects of specific molecules on cell migration through biomaterials.
Another 24-hour experiment. My cell migration data makes no sense. It's way too variable. I'm never going to graduate.
Have you been using a RadWell assay during your experiments by any chance?
I have, yeah.
Well, that's why your results are so variable. In order to detect cell migration, fluorescently tagged cells have to cross a black membrane that can be variable in terms of pore distribution. This pore distribution can lead to significant variability in final cell migration results as cells encounter a different membrane in each transwell.
But how can I get good data?
Well, Monish is going to tell us about our new RadWell insert.
Hey Peter, have you ever heard about the RadWell insert?
No.
Well, it's a device that provides consistent cell migration data as well as assurance for active migration, and it reduces assay runtime.
Nice.
RadWell inserts are designed for detection of radial cell migration. Fluorescently tagged cells are placed on a seating scaffold that aligns with a black dot in the center of an otherwise clear bottom. Cells actively migrating through your choice of a biomaterial will detach from the centered seating scaffold and move radially outwards. Once outside the black dot, the cells can be detected using a microplate reader. So now, instead of measuring cell migration through your biomaterial and membrane, you can just measure cell migration through the biomaterial. This will give you accurate data if you truly want to study cell migration in biomaterials. Also, with these RadWells, you can collect all the cell migration data within 4 hours with easy data collection using a microplate reader. So say goodbye to those 24-hour cell migration experiments and say hello to RadWell.
Yes, no more staying in lab overnight. So how do these RadWells work?
Well, that's easy. Setting up a cell migration assay using a RadWell insert in a 24-well plate format can be done in 4 simple steps. So first, you place your biomaterial of interest inside the RadWell. Make sure you allow enough time for the material to gel, and fluorescently tag cells that will be used in the assay. Second, load the cells into the seating scaffold. Third, place the appropriate media outside the well and the seating scaffold on top of the hydrogel. And last, place the appropriate media inside the well.
Finally, some good data. Now I can finally finish my PhD.